So our last talk comes from Helen Foster at the University of Leeds, and she's going to talk about the multi-step mechanism of axoneme assembly revealed by in situ structural biology.
Okay, great. Hi, everybody. I'm Helen. I'm a new group leader at the University of Leeds, so I started there in August. And I really want to thank the organisers for inviting me to give a talk. I've been attending online for the last couple of years, but it's really nice to be back in person. And so today I'm going to talk about the work I did during my postdoc. And the aim of that was to try and understand the mechanism of axoneme assembly for motile cilia. So I'm just going to kind of start, if this works. No, it doesn't work. Oh, I have to turn it on. There you go. By introducing you to the world of different types of cilia that you have within your body. So we have these motile cilia, which are long hair-like extensions that extend from the surface of cells, and these beat to produce fluid flow. So they're really important in the lining of your respiratory system, where they move mucus and remove pathogens from your lungs. And they also move the cerebrospinal fluid in your brain ventricles. We have the tail of sperm is a very related type of motile cilia, and in this case, it's allowing single cell motility. There are very specialized types of cilia that are only present for very specific stages during development. So these are nodal cilia, and they're only present for a week, and they beat in a slightly different way. So instead of going backwards and forwards, they kind of go in this circular motion. But they're really important for moving morphogens from one side of the embryo to the other, and that's actually what determines the left-right aim symmetry in your body, to make sure that your heart is on your left and your liver is on your right. On top of all of that, we've got lots of modified cilia structures in your sensory organs. So in your eyes, in your inner ear, and in your nose, these are packed full of different receptors, like photoreceptors, and they're really important to help us sense the environment. And then on top of all of that, nearly every cell in your body at some point will have a primary cilia. And these have a very different function. They're packed full of signaling molecules and are really important for individual cells to kind of sense their environment, but they are immotile and they don't move at all. So there's a really, really big diversity of different types of cilia in your body, and they all have very specialized functions. And what's quite amazing is they're actually all built of the same sort of core building blocks, right? So they all are formed of a microtubule-based axoneme, and this gets adapted in many different ways in these different scenarios. And what that means is there's a lot of interest in understanding how all these different structures are generated and how they're different, especially because, as you can see, they're in very diverse sort of parts of our biology. And that means if you have ciliopathies, you have lots of different problems for different unknown reasons. People have diseases that quite kind of variously affect different cilia for different reasons that we don't really understand yet. So during this project, I wanted to focus on how these structures are generated, and I specifically focused on motile cilia. So if we take an example of like a cartoon of the cilia, you can see the microtubules all they're tethered at the basal body, and then they all go and extend towards the tip. And if you cut a cross-section through a motile cilia, you can see it has this 9+2 arrangement. And this is highly conserved, so from human trachea into this model organism, which is the green algae Chlamydomonas, which is where I carried out this work. So they have two different types of microtubules within cilia. There are the 9 microtubule doublets, which are around the edge, and then the central pair of microtubules, which are in the middle. And what's really important is that each of these different types of microtubules are completely coated and covered by unique different proteins, which are the motors and the regulatory components, which together power the ciliary beat. So you can see here they're really coated all the way along these microtubules. They're packed full. Since the early days of EM, we've come a long way. So now we really know the molecular architecture of the different components which actually bind to these different types of microtubules. So this is an example structure that was published a few years ago by the Brown Lab. And so in this single image, there are over 1,000 different polypeptides and over 120 different proteins, right? And they're colored with their different repeats. So they're binding to the microtubules with 8 nanometer repeat or 16 nanometer repeat, all the way up to 96 nanometer repeat, which is the longest repeat that we know about within motile cilia. And so it really is kind of a giant jigsaw puzzle to try and for the cell to try and fit all of this together. And now we know the final kind of what is made by the cell. We can start to ask what are the molecular details of how this structure is generated. And you can think about this kind of as, how is a cell generating this huge cellular skyscraper? Cilia are 10 to 15 microns long, and they can be made in about 90 minutes. So it really is a very fast and reproducible process. And I really like this image of the Empire State Building construction, because as you can see, all of the activity is happening at the top here. There's scaffolding that's being put up, and all of the new components are being transported to the tip to then be grown, to then be put into the growing structure. And that is exactly what is happening within cilia. So the ciliary tip is the exclusive site of axoneme assembly, and that's where the dynamic microtubule plus ends are all localized. And so that really is where sort of all the action is happening. And so what we decided to do was to image the tip by using cryo-electron tomography, and that would really allow us to capture those intermediate states in assembly in situ. And so this is an example tomogram. You can see the tip here is on the left, and then this is further towards the base. You can see all of the different microtubules going in along, and all of the protein components which are bound to their surface. And so you can see the field of view is about 1.5 microns. So we used quite a low pixel size for this, and that was really just so that we could get these kind of early assembly stages right down to what we hope was kind of the much more mature structure. And so once we had this data, we could start to make models and map out our observations. So today I'm actually just going to talk about the doublet microtubules, but we did do a lot of analysis on the central pair, the other type of microtubules as well. And so these were the types of things that we could see. Doublets are formed of a 13 protofilament A tubule, and a 10 protofilament B tubule, which is attached to the side. And in our tomograms, we could see the ends of the A tubules, and these contain plug-like structures, which were observed in the 80s, and they're already known to actually track the end of the microtubule ends during growth and disassembly. So they really do remain attached to the microtubule ends. And then we can also see the B tubule ends, which are more open and lack these plug-like structures. And then we also looked at the furthest extent of the decoration of these outer dining arms. So these odors are the major force generating motor within the cilia, and they're actually what power the sliding between the two microtubules, which then leads to the formation of the microtubule bend. So we mapped out the position of each of these components, and then we could generate 3D models. And so with these models, we could then measure the lengths of different microtubules in different conditions. So for example, we looked at the lengths of steady state cilia, as well as in regrowing cilia and in resorbing cilia. So for the regrowing, we managed to induce cilia shedding by pH shock, and then image them after about half an hour, so while they were in active regrowth. And then we added sodium pyrophosphate, which induces the cilia to resorb by an unknown mechanism. But we looked at the different lengths of the different sections that we measured. So we measured the section here, which is this singlet region that only has the A tubule, the region that has the A tubule and the B tubule, and then the region that has the A tubule, the B tubule, so the doublet with the outer dining arms. And we could see that the steady state and the regrowing cases both had actually surprisingly similar lengths of their different microtubule sections at the tip. And we think this is really kind of demonstrating that the steady state is like a suspended animation, or the regrowing state gets to an equilibrium, which then is held when you're at steady state length. So beyond taking these measurements, we obviously wanted to get into the molecular details of this process. So then we performed subtomogram averaging. And so we so what we did is we took each of these sections and we extracted particles every 8 nanometers along the length. And I said we had 3 different sections, but actually during our analysis, we did classification of this region here, and we found that there were 2 distinct states in this in this region. So actually we had 4 different stages of assembly, but I'll go into the data analysis a bit more in a second. And so this is what we found from the data. So this is now the 8 nanometer averages looking down the barrel of each microtubule, and then this is going to cycle between each state. So from the singlet region down to the base and then back again. And so you can just start to visualize the changes that we can observe within this data. So you can see, we can see changes both in the microtubules and the proteins which are bound to their surface. And we really did learn a huge amount from this data. So I'm not going to go into all of the details. I just want to give you a few of the key points and the main highlights. So the first thing we wanted to understand is what is bound at the very tip of cilia here. So this is the nascent microtubule. So we figured this was the region where anything that was patterning the microtubule would be bound. And when we looked at this structure, we didn't see that many components that were present at the very beginning. But when we looked really closely and compared this to an undecorated 13 protofilament microtubule, we could see these 2 very, very small densities which were bound in the lumen. So these correspond to 2 known filamentous MIPs called FAP53 and FAP127. And these are already known to have 48 nanometer repeat. That means they can pattern the repeat of the axoneme. And they also stick an arm out through the microtubule lattice, and so they can also determine the position of the B tubule growth of the new B tubule. And so you might look at this and think, these proteins are really, really small. How did you do the alignment to actually manage to latch onto them? So that was actually quite a difficult process. So I want to go through actually how we managed to do that. So we did that through some very careful subtomogram averaging and applying of the priors, which are so important to incorporate into your data processing when you have tomography data, to incorporate that biological context. And so what we did is, because we knew that, so in the singlet region at the tip here, we didn't have very many positional markers that could tell us the right orientation of each filament, right? But we did have these further back down the filament. So in this region here, but we have the odors that bound and the B tubule. We know that the A tubule here, it has a seam and it has like a discontinuity at this site. And so when we have the B tubule, we can align all of these particles with the seam and we can find the right protofilament, right? But in this region here, we would just allow free rotation, right? So then we wouldn't have been able to actually capture those densities. So what we did is we took the angles that we defined for these regions and then we applied them to the region further up. And so we did that through careful modeling. And so we just did this manually in iMod. And so we had each filament, we split into 3 different sections. And we made sure that we kind of started all of our models at the tip going down to the base. And so this made sure that all of our filaments had the correct polarity. And then we split each of these filaments into 3 sections. We gave each filament a unique ID that we stored in Reliant star files, in helical tube ID, and then we indexed each of these different sections in different optics groups. So then we could mix together the data and then split them up again as we wanted to. And then from those initial, from the initial 3D model, we got the initial tilt and psi angles for Reliant. So that is the kind of rotation and the tilt, the tilt of the angle of the filaments within the tomogram. And then we ran 1 round of refinement with free rotation around the filament axis, but constraining those first 2 angles, right? And then once we had that rotation angle for these 2 sections, we could then transfer the median angles between these sections on a per filament basis to the singlet region. And then after that, we had the fixed angles and we only did very local constrained searches. Another thing that was really important for our data analysis was the tilt series refinement. So a lot of people will be aware of this, but I just wanted to really show you how important it was for this data. So we did this using M, and I really like the warp M Reliant pipeline. So this is just an example of exactly the same particles before M, after M refinement, and then after M refinement and running one round of 3D auto refine and Reliant. And if you look in this region here, these are the filamentous MIPS, and I hope you can appreciate this process really made the difference between us being able to visualize these and not. So that was a bit more of the data processing side. I want to go back to the biology a little bit and talk to you about the next question that we were really interested in understanding. And that was after this initial patterning of the microtubule, how this B tubule is actually stabilized and bound to the A tubule. So at the very top here, there's a site where the B tubule is nucleated, and that's called the outer junction. And that is done through direct tubulin-tubulin contacts. And then there's a site on the other end of the doublet, which is called the inner junction, and that's mediated by a heterodimer of FAP20 and PAC-RG, and this is called the inner junction filament. So that sits between the A and B tubules, and then the interaction is stabilized between additional proteins on the internal face of the B tubule, which really staple together these two, the protofilaments here, and they're really important for doublet stability. And so we wanted to ask how the inner junction is formed. Do you need all of these components to be present to generate a stable B tubule attachment, or are some of them dispensable and some of them, some of them we don't need? So if we look down now, we're cutting through the inner junction here. Here's the A tubule. And as you play through this movie again, you can see here's the B tubule, and this is the inner junction. And I hope you can really quickly appreciate that it's a very gradual process that it's being built up over time. And actually, so out of this inner junction filament where there's the heterodimer, only one of these components is present at the beginning, only one is required, and that one is FAP20, whereas PAC-RG is really sporadically incorporated over a very long time period during the assembly process. If we look at the internal face of the inner junction, this is what we expected to see. So there's FAP52, which has two WD40 domains, and FAP106, which has like an alpha helix here. And so this is what we expected to see. And this indeed is what we saw when we were down here towards the base. But actually, when we got into this region, region 2, we could see that a completely different structure was bound. So this doesn't have 16 nanometer repeat, it has 8 nanometer repeat, and it doesn't seem to resemble the known canonical components in any way. So this is what we managed to classify out during this section. While we were looking and studying the inner junction, we actually found that about 50% of the particles had this 8 nanometer repeat rather than the 16 nanometer repeat, and all of those particles were much more proximal compared to the ones with the expected architecture. So we've really found a completely unexpected intermediate there. And then the last bit of data I wanted to show you is something that kind of came up during our processing, and a question we weren't expecting to answer even. But we could see during the later stages of assembly that these microtubule inner proteins, these MIPS, they came up very late in assembly. So in stage 3 here, they were absent, and they only came up in stage 4. And this is about 700 nanometers back from the end of the microtubule and the ciliary tip. And so we were very interested in understanding how these components actually managed to reach their binding sites, because this lumen is like this lumen is closed, and as I said, they could be diffusing in from the tip, but actually, like I showed before, the A tubule ends have these plug-like structures, right? So we didn't understand how these components could actually be, even if they were being incorporated from the tip, how they would get to their binding site. And so for that, we looked at the structure of this region here and solved this 96 nanometer repeat of this region. And then when we looked at that, we could see that all of the major known components that we expected were present. So all of the outer arms, all of the regulatory factors, the radial spokes were all present. So it all looked quite normal until, well, it didn't take us that long, but until we noticed there was something quite important missing. So here we found a lattice gap, a gap in the whole of the lattice. And this was periodically placed once every 96 nanometers in the same place. And this was in all of our particles down here. We didn't have to do any classification to try and find this. It really seems to be all of the particles in this region of the tip have this hole. It also has these additional densities on either side that hadn't been observed before. And we think that, and it's also very close to this pink density, which corresponds to a coiled protein, which is known already to be responsible for the spacing of different components within the 96 nanometer repeat. So we really think that these gaps are being intentionally made in the lattice during assembly, and that the removal of tubulin dimers can allow access to the components into the lumen so that they can find their binding site. And so that was the last bit of data I wanted to show. There was a lot more we found out, and I was really lucky to be able to collaborate with Margot Riggy for this. So it's a very complicated process, and she generated an animation that kind of gives a really good overview of the whole process. So I'm going to talk you through that now. So it starts at the very beginning. It starts with the ciliary tip, where we have this nascent microtubule growing, and the plug is tracking the end. And then we have FAP53 and FAP127, which are these filamentous MIPs, which are then patterning the microtubule within the lumen and also determining the site of B-tubule addition. In the next stage, we have the growth of the B-tubule, and this is only needs FAP20 to be stabilized. We'll watch that part again. At this stage, we have the binding of this coiled coil, which is important for the placement of all of the components on the outside of the microtubule, but those large components aren't present yet. On the inside of the lumen, we have binding of components, which I haven't gone into this at all, but we actually think those are really important for modifying the lattice of the A-tubule to kind of compress the lattice and really then promote B-tubule formation. And now if we look on the inner surface of the inner junction, you can see this is this non-canonical intermediate structure, which is present only for a short amount of time. And then this is replaced by the known canonical components, which then really stabilize the inner junction binding of the B-tubule to the A-tubule. And at the same time as this, we have the formation of these lattice gaps. So we see these densities that we really think are recruiting the enzymes to be able to remove the tubulin dimer from the lattice. And we have also at this stage, at stage 3, we have the binding of the regulatory components. So there's one module here, which is formed of the NDRC and radial spoke proteins. And these are actually binding before the major force producing components. So the outer dynein arms are being incorporated after all of the regulatory components. And then the formation of these lattice gaps then allows the components to come into the lumen, and all of the components which are within the lumen can actually are of the right size to be able to fit through this hole. And so only then the lattice is in the lumen is completely coated. And in the very final stages of assembly, we then have the final closure of the lattice gap. We have sealing of the inner junction, and we also have the incorporation of proteins to the inner lumen of the A-tubule, which are actually very species specific. So all of these components are chlamydomonas specific. So we think there's actually kind of the ability to adapt this structure is already built into the assembly process. And so that was what I wanted to talk about today. I just wanted to, I hope you enjoyed the movie. It is available on the bioarchive paper. I just want to recap that we found that the luminal MIPs are really important to establish the DMT periodicity. Another key finding is we found the protein availability doesn't seem to be the sole determinant of axoneme incorporation. So, for example, we see that the tubulin dimer should be very abundant at the tip, but it is removed and replaced into the structure in a very precisely controlled way. We also have the PAC-RG, which is not incorporated from the beginning. That's actually very kind of incorporated in a very sporadic way and much later on in the process, despite it apparently being fully available. And another very important protein, NME7, which binds at the seam. There are 2 copies, but only 1 copy is incorporated before the other. So we think there's a lot of regulation going on in this system. And we also think another key take home is that the emergence of the structural stability and the motility are really carefully coordinated. So we get the building of the foundation of the structure, and then after that we get the incorporation of the motor proteins, and that would ensure that the structure isn't being kind of overly stressed before it really becomes can withstand the stresses of all the motors and the forces which are put onto the filaments. And so, yes, as I said, you can, if you're interested in the mechanism of central apparatus assembly, we also analysed all of that. And the paper also contains lots of other interesting capping structures at the very ends of microtubules. If you're interested in finding out more, please do go ahead and look at that. And I really want to thank everybody who supported me during this project, Gaia and everyone at the Human Technopole in Milan and the funding, everyone who worked on the project, especially Jose, who did some genetic analysis for us, and Margot for collaborating and working on the animation. And yeah, I just wanted to say again that the lab was established in 2025, and if you know a UK-based PhD student that might be interested in starting the advert, is coming out next week. So if you know anybody who's interested in cytoskeletal regulation, please do encourage them to get in touch with me, and I'm happy to take any questions.